Thank you, Martha, for inviting me to be here. And I have to say that the performance art parts of this meeting have been so fantastic that I feel unworthy to be up here to talk to you today. Really fantastic. But Martha asked me to talk about our research and how it applies to the area of personal relevance. And to give credit where credit is due, I had my own title, but this is really Martha's suggested title, so I liked it a lot more and decided to use it. Let me tell you what we're going to be talking about today. A brief introduction to the area of personal relevance. We're going to talk about neuroscience and personal relevance in three areas. Our research on humor, cooperation, and creativity. You might not think that those are things that have clear neuroscience relevance, but I hope I'll be able to convey that to you. And we'll talk very briefly about what I call personalized neuromedicine, which is hopefully revolutionizing how we treat brain disorders and brain dysfunction. And we'll hopefully have a few minutes for questions. So I have an ambitious agenda here. I'm going to go through fairly quickly these areas and just try and give you a taste or introduction. So personal relevance, this is not a term that I have used often in my work or necessarily even in my personal life, but I thought it was an interesting concept, and I looked it up and tried to find how other people define this. And there's some interesting definitions that you can read on the screen. The one I like the best is from a middle school teacher who said, personal relevance is establishing a sense of like-minded ideas through social networking. I thought that was a really good definition. And, you know, our brains are built this way, and when we talk about cooperation, we'll talk about social networking and so forth, but our brains are built to be networked socially around other people, to take information from other people, utilize that somehow in our own lives, and connect with people around ideas, concepts, relationships, and as you've heard, education, vocation, and so forth. But there's a question, and I'm not an old fogey, by the way. I use a lot of technology in my lab, but there is this question that is out there, and I don't think it's answered yet, is personal relevance, as defined by this middle school teacher, potentially compromised by the exponential growth in human technology interaction? So, for example, are these two people interacting in some way? This is obviously a photo I stole from the web, but it's not uncommon to go out to restaurants and see people who are on their smartphones and interacting maybe 70–80% with their smartphones and 20% with those around them. Now, why is this attractive? I'm not going to go into this in a deep sense, but I think it's attractive because not only is it convenient, not only is knowledge available at the touch of a button, but it's kind of easier in some ways to interact with other people. And if you ever look on the web at comments that people make to one another when they post some sort of opinion, it's really easy to insult somebody, to be brief with somebody, and you don't have to dig in and try to decode the social interaction. But I think, in fact, that neuroscience provides us with one way to perhaps enrich human technology interaction, but certainly human-human interaction, and provide insights about human brain function and behavior that are not available from any other source, and bridge apparent differences among us that might be amplified by technology. We like to talk about this in my lab as the dark matter or dark energy of social interaction, all those things that we really don't understand, the 90–95% of social interaction we don't yet understand. So let's talk about humor. I just gave a talk about the neuroscience of humor to a group of psychiatrists and neurologists and neuropsychologists who are interested in this for a lot of reasons. There's actually a lot of clinical implications around the neuroscience of humor we won't have time to talk about, but as you all know, laughter humor occurs in all cultures. It's ubiquitous in infants. I have a colleague who has a young child, Manish Sagar, in the audience, and he's laughing, right? Isn't he laughing at this point? Yeah, at four months, is that right? Yeah, it's wonderful to see that. It's a key component of positive affect as well, positive mood, and a better understanding of humor, we believe, can contribute to a better understanding of brain health. and disorders. Now, we've done a series of studies that have been published very broadly and interestingly have probably attracted more media interest than any other research that we've done, both in children and in adults. And I'm just showing you some of the stimuli that we've used in an MRI scanner in a functional MRI study where we showed funny cartoons and cartoons we slightly altered. These are my favorite ones. This is, in fact, this is my most favorite one right here. There's two nails here, and one says, "Then I heard a loud bang, and when I turned, he had gone." Right there. Okay. A child study used videos from America's Funniest Videos show. Now, I'll give you the punchline, since this is a humor talk, about what we found. Is, in fact, that there's two main systems that underlie the neuroscience of humor. There's what we call a cognitive system that processes the information, the language, focuses your attention, decodes the information, interprets it, and that's part of the prefrontal cortex and so forth. There's an area of the brain that's involved in detecting incongruity. What makes a really good joke? I'm not going to have time, but in my talk I gave in Orlando, I told the audience the world's funniest joke. Okay, if we have time at the end, I'll tell you the world's funniest joke. But what makes a really good joke? You're set up for something, and then there's an unexpected part. That's the incongruous part of it. And that's very, very important. And then there's an emotional part that responds with reward and makes you happy that you're there and so forth. Now, let me tell you the part that's interesting, I think, to us, that there was huge amount of media interest, and it remains. I ignore them at this point. I keep getting inquiries to talk about this because people are so intrigued. And there was a huge controversy, by the way, in the magazine Vanity Fair, where the headline was Christopher Hitchens, I don't know, the late Christopher Hitchens wrote something about why Stanford researchers have proven why women have no sense of humor, which itself is a funny thing because we didn't say anything about that. And in fact, we found that women had the same sense of humor as men. They just processed the information differently. But our main papers in the adult study showed, first of all, that the same reward areas that are activated when you see something pleasant, when you abuse certain drugs like cocaine, are active when you actually see humor, and that's across men and women. But we did see these fascinating differences on the basis of sex. I won't have time to go into it, but men and women do seem to process humor information differently. They found the same number of cartoons funny, the same number. They had the same response. But men and women were really, really different. And we also found that personality seemed to predict how you responded to humor as well. If you're interested in a review where we talk about these areas, we wrote something recently. Two of my wonderful former postdocs, Pascal Vertica and Jessica Black, wrote this with me, and it's in Nature Reviews Neuroscience 2013. One of the fun parts about thinking about this is for example, how do these differences across men and women on the basis of personality come to fruition? How do they arise? Well, we're not sure, but in the end, it's always a biologic push, I can tell you this punchline as well, with the environment moderating that biologic push. Are we set up, are our brains set up to respond to these types of stimuli differently from birth? We don't know that. We found in children that they in fact show the same pattern of sex differences and temperament, the early form of personality differences that we see in adults. But those were in children 6 to 10 years of age. We're going to do a toddler study, and we're going to try and push it down earlier and find out about the environmental effects as well. But one of the fun things to think about is, how did it get this way? Do we actually have a brain that's built from an evolutionary point of view to respond to humor? Probably not. I think humor co-opted these circuits of the brain in the service of something that binds us together, that makes for personal, social relevance. Again, the review goes into this in more detail, but it's a very interesting thing to think about. Advanced brains utilizing co-opting circuitry that was built for something else. Okay, let's talk about cooperation. I'm going to play you a video. And I want you to listen carefully to this. These birds are the common wren in Ecuador, and this is their song. I'll do it one more time because it's actually so beautiful, at least from my perspective. So, do you think that's one bird, two birds, three birds? Sounds like one bird with a lot of trill. It's actually two birds in an intimate interaction, a male and female bird, whose brains are responding in a synergistic way to one another. And this was a paper that was in Science in 2011 from Eric Fortune at my former institution, Johns Hopkins. It was entitled Neuromechanisms for the Coordination of Duet Singing. And these birds have a deep nucleus in their brain where their responses, the cells, the neuronal responses are greater in males and females compared to the male and female syllables alone. So their brains are built to interact, to cooperate, to communicate. And there's some wonderful quotes in this paper I want to read to you, that behavior and neurophysiology show that both sexes coordinate the timing of their singing based on feedback from the partner, And cooperative behaviors are found across taxa, across species, and can be critical for survival and reproduction. To achieve cooperative performances, brain circuits in each individual must integrate both from the animal's own self-generated sensory feedback and from sensory cues produced by the partner or partners. And they went on to speculate that premotor circuits in humans may preferentially encode combined cooperative behavior. Now, isn't that interesting? Because we happen to be doing a study using a form of functional imaging, which is called functional near-infrared spectroscopy, very much like fMRI. It measures the same proxy of brain activation as fMRI. And we decided to, in fact, without knowledge of this paper, measure the prefrontal cortex activity as two people engage in a cooperative activity, okay, in a cooperative game. And in fact, what we found is that there was increased coherence, there was increased correlation between the two brains. This was only, I think, 12 or 13 pairs to start off with, when they were cooperating. And the better they cooperated, the more these two people were synchronized in this prefrontal region of the brain. You would not see this if you were measuring an individual brain across time. It's the correlation of two brains, much like the wrens in that 2011 article. So we thought this study suggested a new class of neurobiological markers that might be used to understand the nature of social interaction in humans. And this is our first paper. This has already been replicated twice in two other groups worldwide. Our current work is built to expand upon this finding, and we have over 100, I think it's 110 to 120 dyads, pairs now, and we're investigating the effect of sex pairings, male-male, male-female, female-female, on this synchronization or lack thereof. And each pair completes 3 tasks: cooperation, competition, and observation of the other person. And we have different conditions, facing and not facing. And with this NIRS technology, as opposed to lying in a scanner and imagining that someone is working with you, which you can do, and which Manish is doing across scanners here at Stanford, you can actually have more ecologically valid research. And we're probing not only the prefrontal cortex in this study, but also an area of the brain that's explicitly involved in social cognition, how you process and interpret information from others in the social domain, called the superior temporal sulcus area. Now, we're at the point where we are analyzing the brain data very carefully, but I will tell you a very interesting behavioral result that we've kind of been spinning around about in terms of interpretation. If you look at, well, first of all, across all the pairings, as the task goes on, they get better and better at cooperation. Okay, so there's a learning part involved. But if you look at the different pairings, female, female, male, 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 female, in fact, when you have a male in the pairing, the rate, the ability to cooperate goes up, the proportion of winning trials. It's not that females or pairings are not cooperating. They're not doing it as well as the other pairings. So you can think about this one. We've looked at the sociology data, the antibody. This is a loaded gun for sure. And we want to be very careful in interpreting this. There are some data, the data are fairly inconclusive, but there are some data in the literature that in certain conditions, you can't generalize, by the way. You see something like this, you read it in the New York Times, you can't say, oh, okay, it's like Christopher Hitchens did with Vanity Fair, but he got it all wrong anyhow. But you can't say, okay, well, women don't cooperate well with women from this. In this particular task, for our study, yes, there was a sex pairing effect. You can think about that one. Let's talk about creativity. Manish has really been the master of this study. I'm not going to spend any time telling this group about the importance of creativity. It is the most, if you look at resumes on LinkedIn, the most widely cited characteristic, although there was an article that accompanied this that said it's getting worn out as well. But obviously creativity is important across the board in virtually all domains of human function and activity. And we've asked several questions. First of all, there's kind of a general understanding, I would say, among our fellow human beings that probably you're either creative or you're not. And the D School here at Stanford, with which we partnered in this research, believes otherwise. And they asked us to be a part of a research study, asked us to design a research study to first of all test if the creative enhancement courses, one of them in particular called the Creative Gym, can actually enhance creativity in anybody. They'd been doing this course for a while, but they hadn't collected any standardized data. We asked the question, what changes in the brain when creativity is enhanced? And is creative capacity enhancement sustainable? Again, getting to this idea of individual differences and how we can understand our fellow human beings' differences from us, potential differences from us. We had some behavioral assessments and neuroimaging assessments. This is the creative capacity enhancement group. In fact, after the training, they were more creative. In fact, the course worked. They were also more focused in a particular way in processing what we call unambiguous stimuli. And they had certain changes in their brains before and after. This was a controlled trial, so we actually had a group of individuals who were receiving the creativity enhancement training, and in parallel, randomized a group of individuals who were receiving Mandarin language writing training, the same number of sessions, the same type of engagement by the teacher, but not the emphasis on enhancing creativity. And then we had them cross over. And so it was a good design, and in fact, brains do change. Not only do you get more creative when you get exposed to this type of course, brains change. So if you ever thought, gee, I'm not creative, or wow, I'm really creative, and that guy next to me is not very creative, it doesn't necessarily, it's not necessarily true, but it doesn't necessarily have to be that way. So these individual differences that we think might be concretized and fixed may not be so fixed. We have other creativity studies that are going on. I mentioned one. We have a three-scanner study that Manish just basically muscled through in a period of about a month with other people in my lab. We've been studying creativity in children and trying to work with Grace Hawthorne in D School to derive and standardize a new method for 21st century method for measuring creativity. In my last 5 or 6 minutes, I want to talk to you about this other topic, which I call personalized neuromedicine. What's the definition? Definition to me is to design more specific and effective prevention as well as treatment for brain disorders dysfunction informed by each person's clinical, genetic, neurobiological, and environmental information. You've probably heard of personalized medicine. Personalized medicine is a good thing. You know, it's been used primarily with genome studies and so forth, but here we have to take into account a lot more, don't we? Because brains are not just shaped by what genes you get, but how those genes determine neurobiology and how that neurobiology interacts with the environment. Let me talk to you first about autism. This is a huge issue in our current society. Those of you, how many of you actually know someone who's autistic or has been diagnosed? Yeah, it's so commonly diagnosed now. It's about 1 in 80, 90 individuals. And there's been a lot of, first of all, I have to tell you, this is my public health message. Vaccines do not cause autism, period, done, does not happen. Okay, so autism is defined by behavioral characteristics. You have problems with social communication, you have problems in your interaction, your social cognition, you have a tendency for having repetitive behaviors. And it can be a very, very, very vexing and difficult and at times tragic occurrence in a family. But what do the scientific data tell us? I'd say up until about 10, 15 years ago, a lot of us in the field, I don't include myself because I have to say, I had a premonition about this 30 years ago plus that we were not going to find a unified cause or pathophysiology of autism. And in fact, we haven't. What we found is that for autism spectrum disorder, there are multiple risk factors. Some of them need to work in combination. Some of them are a little bit more powerful on their own. And there are multiple mechanisms by which you can get to that final behavioral diagnostic state. So the diagnostic term is going to turn out to be so complicated in terms of it being an umbrella that sits over hundreds, if not thousands, of different actual diseases and specific mechanisms. So again, how are these individuals the same? How are they different? Well, they're probably going to turn out to be different in terms of how we address it. So we've taken the tack that let's try to address some of the known risk factors. By the way, when you're talking about genetics, it's always risk. It's never deterministic. So that's my other public health message. Vaccines don't cause autism. Genes don't lead to a single outcome. We know one risk factor is called Fragile X syndrome. It's a specific mutation on the X chromosome, and it leads to a dramatically increased risk for autistic behavior in males who get this mutation. Us poor, genetically inferior males who only have one X chromosome are certainly more at risk for Fragile X, although girls who have this mutation also can show autistic behavior. But up to 50% of boys, young boys who get this mutation will show an autism spectrum diagnosis. It's due to a specific gene, the FMR1 gene, a mutation in that gene, leading to reduced levels of protein of what we call the FMR1 protein, or FMRP. Sounds kind of simple, right? Just give them the protein, they'll be okay. And if we could, we would. Unfortunately, gene therapy for brain disorders is probably at least a decade, if not 2 decades out. Getting genes into the brain is a dangerous proposition right now. This protein actively regulates the translation of a lot of other genes by binding to the messenger RNA and regulating how much protein is produced. So what happens when you don't have this protein in the proper amount? You have all of these downstream effects that are related to other gene products, not just the one from the FMR1 gene. But here's the cool thing in Fragile X syndrome. Unlike a child with autism who doesn't have Fragile X, and for which we don't have a known risk factor right now, who might come into my office, and I am still kind of a real doctor, I do see patients from time to time, I have to treat that child symptomatically. That's the best I can do right now. And it's better with behavior therapy, maybe with certain types of medications in certain circumstances. We can treat those secondary symptoms perhaps to a mild degree of efficacy. We can change the environment. Here's the exciting thing about Fragile X, because we know the gene targets, we know a lot about the downstream effects. We can perhaps work on the mechanisms, alter the mechanisms in a very personalized fashion. The trials of pharmacology are underway. Unfortunately, I think pharma pushed them too fast. They didn't do them very well. They're calling them negative studies. What I call them is unknown outcome studies, because we really don't know. The endpoints weren't very well designed. Okay, now here's our final topic, enhancing cognition. This is a topic of some interest to me personally. Now, as a baby boomer, as I start to enter the stage of known cognitive decline associated with age, we all think that it's good, right? And we are refining our circuitry even as we're in our 50s and 60s. But the fact is there is known cognitive change with aging. So obviously of interest to me personally, as well as from a neuroscientific perspective. What are we doing now for disorders of cognition? I'm going to talk mainly about dementia today, or focus on that. But basically, we have symptom-focused treatments, pharmacological, that are not very good at either preventing or treating the onset of cognitive problems in elderly patients. And there's some symptom-focused approaches like lumosity and so forth. The data are really out on those. We don't know how much those affect change, if at all. But we thought, okay, let's try to introduce a third domain of treatment that involves circuit-focused training. So the individualization comes at the level of trying to help a person gain some level of volitional control over circuitry that could improve their cognitive function. And we do this through a process called real-time feedback. And we use this FNIRS instrument, near-infrared spectroscopy. We're very lucky. We have a lot of wonderful instrumentation that we're using in lots of projects, including this one. By the way, this work is being done by another terrific young researcher in my lab, Dr. Hadi Husseini. And the idea here is that we can detect activity at the surface of the brain for a specific individual and try to enhance that specific individual's cognitive function by giving them feedback about the activity of that circuit that underlies the cognition that we want to improve, or the learning. The results so far are promising. This is a result that is under review at the journal Neuroimage, I believe, for human brain mapping, basically showing that when you get the feedback through this FNIRS instrument, you do better as opposed to getting sham feedback. So that's the proper control, right? You give them the same instrumentation, but you don't give them real feedback about the circuit that you want to improve. And in fact, we see some brain changes that are correlated with this improvement. And this is a small group, but we're thinking about this neurofeedback in a lot of different ways to improve children's cognitive function who have learning disabilities, maybe as a way to improve social cognition in individuals who get the diagnosis of autism, to try and make it more individually specific. So I'm going to conclude by mentioning my key collaborators and funding. I'm not going to go through one-to-one, but one of our funding sources has been MediaX. Thank you very much, Martha and MediaX, for this. We've been very grateful for that funding, and it's particularly allowed us to do our multi-scanner research study that Manish recently concluded. So, concluding remarks. I'm a movie fan, I'm a sci-fi fan. I wanted to be an astronaut. I applied to the program, got through the first phase or two, and then they said, nah. I wanted to be a mission specialist. So I always loved this stuff. I will tell you that one of my favorite movies is Contact. I don't know if you've seen it, it's an old one. It's loosely based on a book by Carl Sagan. There is a scene in the movie Contact where the person who allegedly rocketed off or went off to this other alien world says, I was given something wonderful. This is Jodie Foster playing the scientist Eleanor Airway. Something that changed me forever, a vision of the universe that tells us undeniably how tiny and insignificant and how rare and precious we all are. I believe knowledge is power, And when the knowledge is about the mind and brain and it informs us of our similarities and differences, I believe it's particularly powerful. And my basic premise is that neuroscience has already and will continue to provide novel and useful information about the human condition. And with this improved understanding of our place in the world, we have an unprecedented opportunity to enhance our personal relevance. And I will conclude there. Thank you.